Thank you, Diana. Well, it's great to be here at this second day of the 13th annual LCMS workshop. It's always impressive to see any workshop which continues on for many years, and 13th year is certainly fabulous. So I just wanted to thank Diana for organizing and also making sure that the name of the institute was Renew and not the other alternative that she mentioned. Let me start by thanking the organizers and the sponsors of this workshop. And I was told all the sponsors are accurately listed at the back of this card: Cambridge Isotope Laboratories, SciEx, Wellington Laboratories, Shimadzu, Thermo Fisher Scientific, Agilent Technologies, and Waters. So my role here is merely to introduce the plenary speaker and also to say a few words about the Renew Institute. Renew stands for Research and Education in Energy, Environment, and Water. So we are delighted to have Professor Susan Richardson as a plenary speaker for this workshop, and the talk is also designated as a Renew Innovation Lecture. The Renew Institute seeks to promote interdisciplinary research activities to position UB as a leader in select areas of energy, environment, and water research. We recognize that addressing increasingly complex issues of today requires thinking beyond academic disciplines and embracing creative approaches to solving problems. The Institute's interdisciplinary focus cuts across 7 schools and colleges, including the School of Architecture and Planning, the College of Arts and Sciences, the School of Engineering and Applied Sciences, the Law School, the School of Management, the School of Public Health and Health Professions, and the School of Medicine and Biomedical Sciences. The Institute's overarching goal is to advance energy, water, environmental sustainability as a foundation for a regenerative economy. We will accomplish this via research and education in 5 selected focus areas, which are next generation materials and technologies for energy, environment, and water, sustainable urban environments, freshwater coastal ecosystems and the blue economy, environmental exposures, genomes, and health, climate change, and socioeconomic impacts. And these focus areas cut across the 3 pillars of energy, environment, and water. And when you get a chance, please take a look at our website, which is just, you can get there by simply Googling Renew and Buffalo. So with that, let me now introduce our distinguished speaker. Susan Richardson is the Arthur C. Williams Professor of Chemistry in the Department of Chemistry and Biochemistry at the University of South Carolina. Prior to that, she was a research chemist for many years at the US EPA's National Exposure Research Laboratory in Athens, Georgia. Her research focuses on identifying new disinfection byproducts in drinking water, determining formation mechanisms, integrating toxicological characterization with chemical characterization approaches, and protecting ecological health. She received her BS in Chemistry and Mathematics from Georgia College and State University, and a PhD in Chemistry from Emory University. She is the recipient of the 2008 American Chemical Society Award for Creative Advancements in Environmental Science and Technology, received an honorary doctorate from Cape Breton University in Canada in 2006, and was recognized as an ACS Fellow also in 2016. He's published more than 120 journal articles and book chapters, and writes for the journal Analytical Chemistry on emerging contaminants in water analysis and environmental mass spectroscopy. Please welcome Professor Susan Richardson.
It's a real pleasure to be here, and I hope, apparently the microphone was just working, so hopefully we won't have any glitches this morning. If I was being handed something. But I appreciate the chance to be here. It's wonderful, the real mix of top-notch scientists that we have here, and Diana and Danya have done an excellent job with these meetings. All right, so I changed the title a little bit when Diana said I'd like for you to give this more general audience review and talk. I changed it to Safe and Sustainable Drinking Water. How can we do it? So I'll have, for scientists in the crowd, I'll have normal science, but I'm going to try to give more of an overview. I do have one LC-MS/MS spectrum, and I have a GC-MS spectrum as well, but it's more of a general overview of this area. Okay, so when you think about that glass of drinking water that some of you have in front of you, you might wonder what's in it. So you might have pesticides. It's a rare day that we ever have pesticides that make it into finished drinking water. That's very rare. You might have bisphenol A, especially if you're drinking out of one of those plastic cups that have bisphenol A. A lot of us have those stainless steel coffee mugs and water bottles now. Might have pharmaceuticals, but again, it's a rare day that any pharmaceutical is going to survive wastewater treatment, drinking water treatment, and get in your drinking water, glass of drinking water. And if they do, they're going to be really low nanogram per liter levels. Dioxane. Dioxane is a brand new one on the scene that's really important. EPA has it on the CCL list, the contaminant candidate list now, and Detlef Knoppa from NC State has found it in a lot of waters in North Carolina and also in drinking water. This one's very water soluble, very difficult to extract from water, very difficult to measure. So this is a new one. See, it's very important. It's very toxic. PFOA, perfluorooctanoic acid. That's one that many of us have heard about. We had some, well, at least one of the students had a poster on perfluorinated compounds. Very important. This one, though, rarely do we see in finished drinking water. You see it a lot in river water or wastewater, but very rarely do you get it. Lead. Let's hope you don't have lead in your drinking water, but this probably is not a done deal yet. I've got a slide on this. You probably heard about the Flint, Michigan situation. Very, very terrible situation. I've got a slide showing Mark Edwards' picture for Virginia Tech. He's a personal hero of mine, and he saved the day in Washington, D.C. more than 10 years ago with a very similar situation. So let's hope you don't have lead in your water, but you might. And these chemicals may or may not be in your drinking water, depending. But there's always something that's in your water, always, if you have chemically disinfected water. And by the way, I hope you do, because we want to kill the pathogens to make it safe to drink. DBPs, that's what's always in your water, disinfection byproducts. And these are formed when we add disinfectants like chlorine, monochloramine, that a lot of plants have switched to in the U.S. now, including where I live now in Columbia, South Carolina. Ozone, chlorine dioxide are common disinfectants and very, very important for killing harmful pathogens. We also have UV that's starting to be used in drinking water. And then what happens is when these are used to kill pathogens in water, they're also powerful oxidants, and they'll oxidize natural organic matter as well as unnatural organic matter. But we've got a lot of natural organic matter in our source waters. And this just comes from leaves. Leaves fall in the water, other plant material, other things. It decays. And if you've ever been to a real swampy area, that swamp water can look like iced tea. If you pull it up and you look at it in glass water, it looks like iced tea. The more color you have in that water, the more natural organic matter you have. Totally not toxic, but when it reacts with these disinfectants, it forms toxic DBPs. Bromide and iodide salts can also be in your water, especially if you live in a coastal city where you get saltwater intrusion. And when bromide and iodide are present, they can react with natural organic matter, these disinfectants, to form brominated and iodinated DBPs that are much more toxic than the ones that have chlorine. So there's a concern with this, but you know, to say that 50% of our population lives within 50 miles of the coast in the U.S., so a huge part of our population has saltwater intrusion. And in the world, it's something like 60% of the world's population lives within 60 kilometers of the coast. So this is a worldwide issue. So DBPs are always in your drinking water, and instead of part per trillion levels, low part per trillion, they're usually at part per billion levels. We do have some of these that are part per trillion levels, but usually these are low to mid part per billion levels. So they're a lot higher than some of these other contaminants that we're all concerned about right now. People tend to forget about these. And DBPs can also form from pollutants, so the unnatural organic matter essentially. Some pesticides will have functional groups that can react react with these disinfectants. Pharmaceuticals, same thing, some of them, not all of them, but some of them do have groups that can react. We have antibacterial agents like triclosan. When you wash your hands with hand soap, it contains triclosan. It can react with chlorine, it can react with ozone to form DBPs as you're washing your hands. We have estrogens, hormones that can react, textile dyes, bisphenol A, parabens that have been found in swimming pools, in lotions, and personal care products. Alkyl phenolmethoxylate surfactants can react. It was actually Damia Barcelot's group that showed that a long time ago. He was one of the pioneers at looking at DBPs of other contaminants like this. Algal toxins. So algal toxins are natural. They're not unnatural, but they have microsystems and other cylindrospermopsin. Other algal toxins have groups, functional groups that can react to form DBPs. So if you're monitoring an algal toxin and you're disinfecting your water, you think, oh great, it's disappearing, right? It's disappearing. Well, things don't usually disappear. They transform into other things, and it might be that you've got a DBP in your algal toxin. So and then we also have musts that are fragrance compounds that are in a lot of our personal care products, maybe our laundry detergents. Those things can react too. And a few years ago, this was back in mid-2014, I wrote a review together with Christina Postigo, who's a Marie Curie fellow working with me, published on pharmaceutical DBPs. And I want to just point out a couple of these to you. One I'm going to show you in a minute. Iopamidol is an X-ray contrast media that gets into source waters. It's one of the highest levels, probably the highest level pharmaceutical that comes from wastewater into surface waters at part per billion levels typically. So this is a rare one that gets out there in part per billion. In fact, Thomas Turnus in Germany has found this one and other ones up to 100 parts per billion in some German creeks and rivers. So this is one that has a chance of causing some problems. But what happens is it's totally safe by itself, but when it reacts with chlorine, water, or wastewater treatment, it can form the most toxic DBP, acetic acid, which has also been shown to be tumorigenic in animals. So this is where you can have some trouble. Another one I want to point out is kind of interesting is azithromycin. I think that's still, it used to be the #1 antibiotic used in the U.S. is Z-Pak. If you ever have your kids get sick and get the Z-Pak, it's happened for me. I've taken it myself. This azithromycin can react with monochloramine that a lot of our plants are using now to form nitrosodimethylamine, which is a very potent carcinogen at really low nanogram per liter levels. So this is another example. So if we didn't have any health issues, I would not be too concerned about these things. But this is what we've seen. There are human epidemiologic studies that show a risk of bladder cancer, also miscarriage and birth defects. And this has been seen in several locations, especially the bladder cancer. Not in every city they've studied, but in a lot of them in other countries beside the U.S. as well. Just last year, Mike Wright at EPA, he's an epidemiologist at EPA, in Cincinnati found cardiac defects, birth defects essentially that are cardiac defects in babies. So this is something EPA is taking a hard look at right now. This is a new thing that's been found. And I want to point out, this is kind of hard to look at, but there are other issues that are maybe less major than cancer and miscarriage and things like this, but some people suffer from these horrible skin rashes. There are also some respiratory issues when people take showers, but this seems to be a sensitive subpopulation. It seems to be in the thousands of people rather than the millions of people. This was happening to all of us with chlorinated drinking water, only showering on chlorinated water. Then EPA would recommend not chloraminating. It's not the millions of people, it seems to be the thousands of people. So this is another issue. But I feel for these people because you get the water you get, right? You turn your faucet, you have no choice of what they're disinfecting with. If you happen to be one of these people, then you get stuck with these issues. A lot of them have had to bring spring water into their homes, this big giant bottles of spring water. This particular woman in the Bay Area, in San Francisco Bay Area, decided to take out a membership at a YMCA about 30 miles down the road where they were still using chlorine. Rash went away. I've heard a lot of these before and after stories, but again, it's not everybody. It's just some people that are sensitive. So lead, I mentioned before, is another really important issue. So the switch from chlorine to chloramine, as I mentioned, lots of plants have switched to chloramine. And by the way, let me tell you why they've switched. I'll show you the regulated DBPs in just a minute. But trihalomethanes that you've probably heard about, those are the first DBPs identified back in 1974. Also haloacetic acids. The regulated ones are chlorinated chlorines and bromines in them. And if you switch to monochloramine to chloramine disinfection from chlorine, you'll knock those down by as much as 90%. And so if you're a plant, you're struggling to meet the regulatory limit for those compounds, all you have to do is add ammonia to your water, to the chlorinated water, form monochloramine, knock those down. Hello? All right, we'll try that. And if it doesn't work, then I'll use my teaching voice. I'll get into the back there. So anyway, so that's why a lot of plants have switched to chloramines, because it knocks down the regulated DBPs, and it's cheap. It's cheap to switch to. But what happens when you do that, you can also cause a lead issue. There's several issues with chlorination. The lead issue is where you have, over a long period of time, and this happened in the Washington, D.C., where you have, like if you have a distribution system, pipes that have been there many, many years in D.C., more than 100 years. Where I am in Columbia, they have really old pipes too, they've been there for a while. And just the little bit of lead that's in the water naturally, just tiny bits, will build up in scales inside the pipes. If you've ever seen a water main break on TV or in a picture in the newspaper, the pipe is like this big. And the amount of water going through might be this big. They're biofilms, but also these scales, lead scales. And lead scales are totally fine. They sit there minding their own business. They're not causing anybody any problem. They're insoluble lead scales. But as soon as you switch over to chloramination, chloramines changes the redox potential of the water, and you turn the insoluble lead into soluble lead. And this is what happened in DC and exposed a lot of people to really high levels of lead for 18 months, much longer than happened in Flint, which is really terrible. So this is when it happened, 2001, 2004. Consumer was exposed to really high levels of lead, 48,000 ppb, and the regulatory limit, the action limit is 15 ppb. So that's what happened. Children, Mark Edwards has done a study of children in the DC area, and the IQ levels are lower. It's really pretty devastating. We know lead, right? We know the toxicity of lead is a neurotoxin, so there's no question about what it does. And thank goodness for Mark Edwards in that situation, because it would have gone on a lot longer. So here's Flint, 2015, 2016, and by the way, it's just been recent that they've got the lead under control, because once you flake off and you corrode the pipes and you're leaching everything off, it takes months to get it back stabilized. They've added the orthophosphate back, but it takes a long time to coat those pipes again. So they finally got it down less than the 15 ppb now, but it's been a while. So it's not instant, to fix this problem. And so you think you would have learned our lesson before, right? And Mark goes around, we actually had him give a seminar. Here's a picture of him. It's a great picture inside a pipe. He gave a special lecture at our university at South Carolina last November. It was just amazing. It was standing room only, and he's just a hero in helping lots of people. And it was one of the moms, by the way, I don't know if any of y'all saw, they had a really great show on Nova just about 2 or 3 weeks ago. I just happened to turn it on. I didn't know it was coming on. And of course, Mark Edwards was all over it, and Amy Creedon. Can you give me the back if I do that? No. Okay, sorry. Okay. Or if I can see how this is going to go right in the face. Yeah, maybe that's it. All right, we'll try this one. So anyway, so Mark saved the day. It was Leanne Walters, one of the moms, that 2 of her kids were really suffering with health issues in Flint, and nobody believed her. The water treatment plant said it was okay, water's okay, and it's like red. You'll see pictures, right, and stuff floating in it. Of course, you can't even see the lead. If not for the iron and the other stuff, corroding off in that water, they would have never known. You don't know when lead's in there. So anyway, the Michigan DEQ, Department of Environmental Quality, didn't believe her. They said the water's okay. And it was just to save a few pennies, save a few dollars, and they switched from Lake Huron water, which is, the Great Lakes are pretty pristine as drinking water sources. You don't want to switch away from a pristine source if you've got one. So Flint was buying wholesale water from Detroit, that paying a little extra money to have it piped down to them. But what do? Just to save a few dollars, they switched to the Flint River, highly corrosive. It has 8 times the level of chloride in it than normal waters do, normal rivers. And what do? On top of that, they stopped adding the corrosion inhibitor. Switched to a corrosive river, and they stopped adding the corrosion inhibitor. And as I told you, it takes a long time for that corrosion inhibitor to coat the pipes again. And so really stupid. I mean, Mark said that even if they just kept adding the corrosion inhibitor, they might have been okay. So, but thank goodness Leanne Walters, one of the moms there, I appreciate this, a mom myself, that taking care of fighting for your children, thank goodness she called him fairly early on. This could have gone on as long as it did. And so back to DBPs again, which is the main topic of my talk. There have been more than 600 DBPs identified now, and this has been a little bit my life's work to try to uncover the missing ones, because EPA's regulates, I'll show you in a minute, 11 DBPs, 11, that's all. And for a long time, everybody's just measuring those same 11, or even just the THMs and haloacidic acids, and everybody's ignoring everything else. It's like, there's other stuff there. What else is there? So this is what's there. Whole lot of halogenated compounds, including these halobenzene that were just identified recently by Jing-Fong Lee at the University of Alberta. So even when we think we've got all the classes, there's still new ones that come on the scene. And we also have, let me see if I stand over here a little bit, I don't know if it helps, but some of these have nitrogen in them. They call them NDBPs, or nitrogen-containing DBPs. And these NDBPs tend to be more toxic than the ones that do not have nitrogen in general. These are the ones we regulate. 4 trihalomethanes, 5 haloacidic acids, bromate, which is an ozone byproduct, and chlorite, which is a chlorine dioxide byproduct, and that's it. And there's far less known, little known about the occurrence and toxicity of the unregulated DBPs. And the real rub, the real reason, one of the reasons I'm still in this, is that none of those regulated DBPs, none of those cause bladder cancer in animals. So most scientists think we're missing the DBPs responsible, and it's probably not these regulated ones. And if these regulated ones controlled all the other ones, if they were good surrogates for everything else, we would be fine. I'd be totally happy. But in fact, they're not. And I'll show you that many of the more toxic DBPs are inversely related to the regulated DBPs. So you do things like switch to chlorination to lower the regulated DBPs, increase the formation of some of these more toxic ones. And brominated and iodinated DBPs are also important, I mentioned before. And this shows the cytotoxicity is in the blue color, and the green is the genotoxicity. This is a mammalian cell assay, in vitro assay. That's Michael Pleva's work at the University of Illinois. He's done a tremendous amount of work to point the chemists like myself to the DBPs that we need to be studying. Out of all these almost 700 we've identified, which ones are the most important? Which ones do we need to go after? And ultimately, of course, my goal is to find out how they're formed, find out which ones are the most toxic, identify them first, find out which ones are the most toxic, and get rid of those ones that are toxic. That's my whole goal. And so what we see here is that as we go from chlorine-containing DBPs to bromine-containing ones to iodine-containing ones, we vastly increase the toxicity. This is a log scale. I always tell my students, look at the y-axis. It's really important. Don't miss the y-axis. It's a log scale. So these are big differences here, whereas the chlorinated ones, not all that toxic. Brominated ones, pretty toxic, and iodinated ones, the most. Oops, sorry. And if I had to list which ones I consider to be emerging DBPs, we have emerging contaminants, so I would list emerging DBPs that are unregulated. These are the ones I would list, and it's because of the toxicology and because of the levels we see them at. The halonitromethanes, like this dibromonitromethane, we found this one up to 3 parts per billion. And a common theme with a lot of these is that plants switching over to other disinfectants, we call them alternative disinfectants beside chlorine, like ozone, they increase in formation. So again, utilities, water utilities, responding to the EPA's regulations, trying to meet those regulations, and that's very important. That's what they've got to do, is meet the regulations. Unknowingly, they're actually increasing the formation of some of these. So we also have these iodinated trihalomethanes and iodoacids, and we discovered in my lab these iodoacids just a few years ago in a nationwide occurrence study we carried out in the US, like iodocetic acid, which is the most genotoxic of all DBPs. It's also tumorigenic, and it's increased with chloramination. We found out that these iodinated compounds are vastly increased when plants switch to chloramination. Haloamides are also increased with chloramination. That can be up to 14 parts per billion. These can be really high too. Iodo-THM's up to 15 parts per billion. We found in one of the cities in the US we looked at. But these are pretty high levels, and nobody hardly even ever looks for those. We identified those a few years ago. A few groups are looking for them now, but hardly anybody monitors for them. Halofuranones like MX, very mutagenic. It's also carcinogenic in animals. This is an important one too. Acetonitriles, and there's another NDBP, nitrogen-containing one. These are all unregulated, and these seem to be driving a lot of the toxicity of the mixtures of drinking water we're looking at, and can be up to 41 parts per billion. So these aren't low part per trillion, these are part per billion ones. Nitrosamines, this is a little bit of an exception to that. The highest that we've seen in the unregulated contaminant monitoring rule that EPA has, where it forces drinking water utilities to measure for about 25 chemicals. NDMA was on that list, nitrosamine. This is what it looks like. You see, it doesn't have any halogens on it, right? No halogens, but it's still very potent. It's a carcinogen, and its health reference level for cancer is 0.7 nanograms per liter, 0.7 parts per trillion. So sometimes low part per trillion levels of chemicals can be important. We found as part of the UCMR that data that more than 40% of the US population was exposed to well over that level, that health reference level for cancer. So that's why EPA is considering regulating that NDMA and also the nitrosamines. We have halobenzoquinones that are fairly new DBPs that have been identified. We have only a little bit of toxicity information on them now, but even whole new classes, and we've identified a couple of new classes that don't know this yet recently. This shows the genotoxicity of iodocetic acid, and here's a picture of Michael Pleva, University of Illinois. He has really been a key person in advancing this. As chemists, most of us are chemists in this room, we can only do so much. We can do our chemistry, but it's really, I think it's really important, and they were new at Center hearing that it's multidisciplinary. It encourages multidisciplinary research. This is where I think it's so important. I can identify chemicals all day long. I can study how they're formed, but how do I know which ones are the ones driving the human health effects? So really important to interface with these toxicologists and also with epidemiologists too. I've been working with, so that's another important thing to do too. And engineers, I'll show you a good story at the end of how we're trying to get rid of some of these things and being somewhat successful. But this is in mammalian cells and Chinese hamster ovary cells on the left. Here's human cells, a new human cell line that can be used that has cell contact inhibition on the right. Here's iodocetic acid, bromochloro. So the further you go to the left, the more genotoxic the chemical is. It causes DNA damage. This actually causes double strand breaks to a DNA. It's difficult to repair, so it's a really kind of potent form of DNA damage. But iodocetic acid is about twice as genotoxic as bromo, and then this is more than an order of magnitude more than that for the chloroacetic acid. So we've got iodo, bromo, and then chloro. And then you look at the human cell line, this is amazing. Iodo, bromo, and then chloro. And if Michael was here, he would tell us, this is telling us something. These patterns don't occur by accident. And in fact, he's got a hypothesis with a mechanism how he thinks this is causing toxicity. He thinks it's binding to GAPDH, one of the enzymes that we need to produce ATP in our cells. And if you block the ATP formation, you form reactive oxygen species, ROS, and thinks that's how it's causing the toxicity. Got some data for that. So he has this vast database. This is a little bit dated, this graph I'm showing you here. Rather than showing dose response curves, where you get the full curve, it would be a jumbled mess if we put them all on top of each other. So what he did was he took the of a dose response curve, like the midpoint, put that on the x-axis here, and he calls this lollipop graphs. These look like lollipops, kind of. So the heights mean nothing. They're just for the classes of DBPs. These are the haloacetic gases here. We have the halonitromethanes here in red, haloacetonitriles, haloamides, and here's MX, the compound that we know is mutagenic, very mutagenic in bacterial cells. This is in mammalian cells here, and it's a carcinogen, the animal carcinogen. All these DBPs to the left of it are vastly more genotoxic than MX. And the positive control, ethyl methane sulfonate, is way off to the right. So this is the positive control of all these DBPs. Most of these that are unregulated are much more toxic than either of these. Sorry, forgot to show, here is iodocetic acid all by its lonesome out here. It's the most genotoxic of all. And here's bromocetic acid, which is regulated, but almost never found in drinking water. But we see this one all the time in chlorinated water. In fact, in my own drinking water in Columbia, South Carolina, unfortunately. But it used to be we'd have to ship some waters in when I was in Georgia, ship them in from Texas and other places to get these iodo DBPs. I have them in my own drinking water. I can turn on my faucet and don't have to ship them by FedEx. But unfortunately, I have filters on my water at home. I don't want to drink these things in my water necessarily. And another, what we found is, in addition to iodide salt, I mentioned bromide and iodide coming from saltwater intrusion, in addition to that being a form of a precursor to iodo-DEPs, these toxic iodo-DEPs, we discovered that these X-ray contrast media used for medical imaging are another source. I saw a talk that Thomas Turnus gave at a Lake Louise tandem mass spectrometry workshop a few years ago, and I saw, oh, they've got iodines in the structures, and she was showing these found in source waters at really high levels. I mentioned up to 100 ppb. And what happens is you get these things for medical imaging for soft tissues, your lungs, your kidneys, your bladder, things like this. Anything soft tissue, you get in the IV. And in the IV, you can have up to 200 grams per person per day. Huge amount in IV. Totally safe, though. It just provides a nice contrast for your soft tissue X-ray. Screen it out it goes down the toilet, out it goes to your wastewater treatment plant. Wastewater treatment plants in the U.S. remove nothing of those, zero. That's why there are such high levels in the environment. In Europe, they do a little better job than we do. They remove about 20%, still got 80% of it coming through to the surface waters. And if you happen to be downstream of a plant that's released as a wastewater plant, you're downstream, the city downstream, your drinking water supply might have this come in, and then you treat with chlorine or monochloramine, and you have natural organic matter present. You can form these toxic iodo-DBPs, including the iodo-THMs and iodo-acids. We've also used LC-MSMS to identify the high molecular weight things that are formed initially from these compounds. And this is one of the brand new ones we've identified, or two brand new ones really, iodocetonitrile and chloroiodocetonitrile. This is a GCMS chromatogram. We're using high-resolution mass spectrometry. It's my student Christina Joseph working on this. So what we've done is, once we discovered iopamidol was in a lot of our source waters in the U.S., we started reacting them in the lab to understand, are they sources of these iodo-DBPs? So we're taking river water that contains natural organic matter, reacting with iopamidol, the one that we're seeing the most often. We measured for several of these. This is the one we saw most often. Turns out it happens to be the most reactive as well. We're forming these DBPs, but here's two brand new ones, iodo and chloroiodocetonitrile, not known before. Here's one mass spectrum I want to show you. And so it was mentioned before, high resolution is so important. We've been doing high resolution mass spectrometry for a long time with GC mass spec, and we're doing some more LC mass spec now. But you see we get the decimal places on these, so you know exactly how many carbons, hydrogens, iodines, nitrogens, whatever you've got in your molecule. And so we know the formula, you know it has to be this formula. You see the nice little 1 chlorine pattern. You know this 1 chlorine. By the way, iodine is 5 stealth. Iodine only has one stable isotope, the 127. So unlike chlorine and bromine that give these nice little patterns, iodine gives you nothing. And except for occasionally you get the 127 show up where iodine gets broken off and you see the presence of it. It's not like it jumps out at you and you and we also recently confirmed that with a synthetic standard. We actually synthesized the standard. A student in my group did that. So I want to mention new issues that we have that I want to show you, new impacts. We've got hydrofracking, wastewaters, produce waters being released in our surface waters and then put deeply injected into our groundwaters. Coal-fired power plants are introducing contaminants as well. We have climate change, of course, and I love that, Environment Canada is now what, Environment Canada and Climate Change, or is that what it is? Yeah, so I love that. So there's no denying it. Canada's out there. So obviously we've got climate change. It's here to stay too. I just pulled up last night the most current drought monitor, and we're actually okay for the moment. Some places are not okay, but this is actually a good picture for the moment. But y'all know we've seen things like this, situations like this, and even where I used to live in Georgia, there was a 6-year drought we had. In fact, I'll show you a picture of that. I don't know if you remember Lake Lanier that serves a lot of the city of Atlanta. They're just pumping. This is what it looked like. This is a boat ramp, and there's docks that are on dry land. It was just shrinking during that long 6-year drought, and finally the rains came and filled it back up again. This is very dire enough that they were trying to steal back the Tennessee River from Tennessee. Apparently the old state lines, Tennessee River used to be in Georgia, and they were trying to get it back so that they could have another water source. It's crazy. We have growing populations, we have water scarcity, so we're looking at new water resources, including wastewater reuse is a big one, and we've got a grant looking at this now, looking at treatment of wastewater, reuse of wastewater. Desalination's starting to become more popular. San Diego just built a new one. Tampa's already had one. This is, and then back to that, I want to show you an example of what we're doing with hydrofracking. This shows all the North American shale plays, the shale gas that we have, shales we have around. So it's not only in the U.S., not only in the Marcellus Shale. We tend to think of the Marcellus Shale, the big one. This is the largest one. But here it is got it in Alberta and Canada, different places. The one I've got some data on, I'm going to show you, is from the Barnett Shale in Texas. But there are lots of shale plays around, and the U.S. is going after these hard. And so a lot of people are concerned, and rightfully so, about what chemicals the companies are putting into the grounds, because they use surfactants, they use biocides, they use other proprietary stuff that they don't even have to tell EPA what they're putting in the ground, and it changes from month to month depending. They can put whatever chemicals it seems they want to. But what I'm a little bit more concerned about, and it ties in with my work, is what happens if those get into drinking water supplies, because my concern is these toxic brominated and iodinated DBPs, and I became concerned when Jean Van Briesen from Carnegie Mellon University started doing some work with looking at trihalomethanes only at first, and she saw huge levels of bromoform, 50 parts per billion of bromoform in a real drinking water plant near Pittsburgh off the Monongahela River when they were doing surface water releases of these fracking waste. 50 parts per billion of bromoform is like more than 10 times the highest we ever see in the U.S. And so I'm thinking, that's trouble. That's a cancer cluster waiting to happen. And what else is there? Bromoform's there. What else is there? These other DBPs. And so we've got collaborators, Jean Van Briesen's working with us, but we also got collaborators in the Barnett Shale, where they're looking at also trying to purify this fracking wastewater. So here's the produced water. So after you inject high pressure water, sand, all this other material into the ground to break up the shales and get the gas oil out of there. And after a while, this is what the produced water looks like that you're going to get rid of. And the Barnett Shale, they're very water scarce. In that part of Texas, they don't get a lot of rain. So they don't want to just keep depleting their water supplies, their surface water, groundwater for doing fracking. So they're trying to clean it up. So here's a pre-treated with a 2-micron membrane, with a 0.1 micron, and here's a nanofiltration with a 1 nanometer membrane. You see it looks pretty nice, doesn't it? It looks pretty clean, so they can reuse that again. But what you don't see is that the bromide and iodide extremely high, I mean high ppm levels, and they're all still in there. The nanofiltration does nothing to remove the salt, so that can get out in the environment and cause toxic DBPs. And this is what we're seeing in some chlorinated fracking water that we're doing in the lab. We're seeing brand new series of iodophenols that we've never seen before. We see where N equals 1 iodine. Oops, I have errors on this one. We have 1 iodine, we have ones with 2 iodines, and we have ones with 3 iodines with GC mass spec that we're looking at. And we also saw these compounds, these new unknowns. These are unknown unknowns that were either going to be these iodobenzoquinones that have never been found before, that other chlorobromobenzoquinones have been found. In fact, Hannah, my student working on this, was all excited thinking, we found the first iodobenzoquinone. How cool is that? You know, it's kind of sad when you get excited about identifying new things, but it's better to know than not know. Better know what's in there. But we realized that these could also be iodocrisols. Looking at the mass spectrum, the full scan mass spectrum, it could also be iodocrisols, which is nothing brings here. And then you see these are very different structures. This one has 2 oxygens. This one has only 1 oxygen. And you see the exact masses of these accurate masses are very different. They are different to that second decimal place. So with high resolution mass spectrometry, we can nail which ones these are. And in fact, what we observed was masses that matched the iodocrisols and not the iodobenzoquinone. So Hannah was a little disappointed, but this is how we knew they had to be these iodocrisols. We have found a whole bunch of these, and we've been confirming a lot of them with standards. I think it's really important to confirm ones that you can, and sometimes you can purchase them, sometimes you have to make them. You can't always make everything, but we only have guesses. We only have tentative identifications until we confirm with standards. And now I want to show you another project I'm working on is on wastewater reuse for potable drinking water. And they're taking a hard look at that again in California. And there are a couple of places in Texas that are already doing this, already been doing this. In fact, El Paso, I just visited there not quite a year ago, where they broke ground on a direct potable reuse plant. They've been doing indirect for a while, just like Orange County. Now El Paso's in the middle of the desert. They've got salty groundwater. They're very water stressed. They've got to do something. So what we're doing in the studies, we've taken 21 of the most prevalent wastewater contaminants that survive wastewater treatment, ones that are at the highest levels, and also ones that are present at levels that can cause toxicity or other effects, either on humans or on wildlife, or on fish and wildlife. So we've taken and got this list of 21. There's a couple of priority lists from the state of California. Looking at those, BPA, bisphenol A, is one of those on the list. And so this just shows, just to want to illustrate, this is with a LC-MS chromatogram, where we're looking at bisphenol A DBP. So of course, where we're looking at advanced oxidation treatment, hydrogen peroxide and UV with hydrogen peroxide, like they're doing in Orange County, but also looking at effects of chlorine once you treat that water with chlorine, which is what's going to happen for drinking water. They're going to treat it with a disinfectant. And so we found that you can form these chlorinated BPA, bisphenol A, and this has been seen one time before. A couple of these have been reported in another group before us. This is using high-resolution mass spectrometry with the LC Orbitrap that we have in my department. And this is tetrachlorobisphenol A, showing the accurate masses that we're able to tell exactly that this is what it is. And we see the theoretical, and then the observed is good to that 4th decimal place, in this case with higher resolution. Okay. And oh, wait a minute, I just showed this a second ago. I'm sorry how that snuck in there. Sorry. That one I showed you just a minute ago. So the last thing, very last thing, I hope I'm doing okay on time. I'm running over on time. Okay, all right. So so I've shown you all these new things we found in drinking water. It's very important to uncover them and all these new issues, these new impacts on our drinking water. But then what can we do about it? So, as a chemist, I can't do much about it, but working with engineers in this case, I can do something about it. And so I'm working with Detlef Knappa right now from NC State on a grant from the Water Research Foundation, along with Scott Summers at University of Colorado and Eric Dickinson at Southern Nevada Water Authority. And we're looking at using GAC, granular activated carbon, to remove the NOM precursors, the natural organic matter. So the idea is if you knock the precursors down, you should be able to knock these DEPs down. And so, but plants used to think this is just too expensive, we can't do it, too expensive. I think it's just pennies on your water bill. It's not that big a deal. I'll show you a picture. Cincinnati, city of Cincinnati's been doing it for years. But it's kind of back on the table because people realize there's all kinds of problems with switching disinfectants, like I've shown you, switching to chloramination in particular, and they're thinking, in EPA and the regulatory office is thinking, no, maybe it's better if we go back to square one and just try to remove the precursors to lower DEP levels and make it safer. So the GAC looks like this. It has all these pores in there. And it's similar to the material you have in a Brita filter or your other under the sink filters. You know, it's activated carbon, looks like that. So just on a larger scale, so you've got your Brita filter, and here's kind of what it's like in a drinking water plant. So it's just like a big Brita filter essentially. And I should point out Amy Cuthbertson, one of my other PhD students. This is her main project. She's fabulous. And here's what it looks like in real life. It's a little uglier in real life. This is the one in Cincinnati. And you know, the reason they've been using it for so long is because, does anybody know what river is their water source in Cincinnati? Who knows that? Anybody know? Yep. Ohio River, that's right. Yeah, because Angela's in Cincinnati, EPA in Cincinnati, Angela Bat. So Ohio River, you know, not so pristine, right? It's not like Lake Huron or Lake Erie even. It's, you know, it's gone through seems like every major industrial city in the Northeast by the time it gets to Cincinnati. And so they can have as high as 8 milligrams per liter of dissolved organic carbon. And I can tell you that is not natural. In Athens, Athens, Georgia, and even South Carolina, where Athens, Georgia, we had levels of 2 milligrams per liter, and that's a warm climate. We have a lot of plant growth, a lot of leaves falling in the river. They got 8 in Cincinnati, so that's unnatural organic matter. They're trying to get rid of all those industrial pollutants that are coming down the river. And so Amy created a method, along with my postdoc Susanna Kimura, a very ambitious method for about 60 priority unregulated DVPs we want to measure. We want to see, there was some information, some data showing that GAC could reduce trihalometane levels, but nobody really knew about other DVPs. And so, you know, the story before, you reduce the regulated ones, these other ones pop up. I want to make sure we don't have something like that here with the more toxic ones. So this is why the interest in this. So we had the iodo acids that are very toxic, the haloacidaldehydes, iodo-THMs, haloketones, halocetonitriles, halonitromethanes, haloacid amides, all these really important DVPs that are unregulated. And these methods we did were built upon methods developed in a nationwide current study that I led back with Stuart Krasner, and we published back in 2006. But in that study, we had low part per billion detection limits. That's all we needed for that study at the time. Now we wanted to get low part per trillion level detection limits. So this is tribute to Amy and all the hard work in getting methods to really low levels, because we want to see removals. We want to, with GAC, we want to be able to go really low, see how much are they removed in drinking water. So just a quick note about the different methods. Unfortunately, there wasn't a one-size-fits-all. Y'all that do, especially W. Shoemaker, doing nice methods at EPA in Cincinnati. Sometimes you can't just do one simple method for everything. You have to break it up sometimes. And so with these 60 or so DVPs are very different chemically. And so the more volatile ones, we could only do a liquid extraction once with MTBEs, similar to EPA methods here, using high-resolution mass spectrometry, GC mass spec, selected ion monitoring, GC mass spec here. Here for these other less volatile DVPs, we could do multiple liquid extractions. If you remember from animal chemistry, 3 times extraction, you get more out. So we're trying to get as much out as we can, get low levels. That's what we're doing for these. And then for those pesky little mono and dihaloaldehydes, they are difficult. They're almost impossible to extract from water is the problem. So if you have chloroacidaldehyde, just like formaldehyde, it wants to stay in that water. So we have to derivatize with this pentafluorobenzyl hydroxylamine to make those things nonpolar, so then we can extract them out of the water and measure them. So I hate derivatizations, but there's not a way around that one as far as I know for now. And this is some of the results. So I'm going to show you a couple of slides of results. Will be my last slides to show you. This extensive DVP quantification allowed us to assess removal of that NOM with GAC to see if we can make the water safer. And also we're investigating pre-chlorination. In this case I want to show you, this is from a pilot plant. We had a pilot plant, several full-scale plants in the study. We're at the very end of the study. So if we just chlorinated that water, that river water straight up with no GAC on the front end of it, we get about 170 parts per billion, all these total DVPs that we're quantifying here. If we chlorinate, or if we use GAC before we chlorinate, trying to knock that organic matter down, knock the DVP levels down, you see we do get really good knockdown, about 50% or so less DVPs if we use GAC. But if we put on top of that, we have a short pre-chlorine contact time of 30 minutes or less, we knock that down even further, which is really kind of cool. So there's what's being knocked down with just the GAC now with a pre-chlorine before GAC. And this work with the pre-chlorination builds on work of Zhangru Zhang, who's at the Hong Kong University of Science and Technology. He's been looking at pre-chlorination and has a mechanism why he thinks it's benefiting, because it's almost counterintuitive. You think, okay, we're using GAC, why would you use pre-chlorine to form DBPs already? So this is what granular activated carbon looks like to the eye. Here's Zhangru over here. And as I mentioned, GAC has all these pores inside, right? You can sorb your carbon on the outside of the carbon, and think about like a Brita filter again, you're sorbing stuff, right? You can also get stuff down in these pores. And so what happens is if you have that short chlorine contact time, you break up the giant natural organic matter. These are humic acid molecules, fulvic acid, big molecules can be 3,000, 10,000 molecular weight. You break them up into intermediate aromatic DBPs, so the big natural organic matter is just going to still stay on the surface, can't get in the pores, but these smaller kind of chunks that you've broken them up into with the chlorination can then more effectively get into those pores and get removed. That's what we think is happening. And here's the last one I'll show you on the impact of GAC age, the age of that filter. So if you think about your Brita filter, you've got it brand new, brand new is going to remove more stuff, right? Because it's all the sites are free, they're not all sorbed up yet. And then at middle-aged filter and then old filter, when it's time to get rid of it, what happens in a real plant? And so this is what we found. Here's the no GAC again when we just chlorinated it. We have not quite 100 ppb of total DBPs. Here's our youngest filter, and you might expect it would do the best, and it did here. And look at this, this is amazing. It's like 80% or so reduction. That's what I like to see. I want to make this drinking water safer. This is good. Here's our middle-aged ones, and not doing as well at this point. Some of the NOM's breaking through, right? So we're forming more DBPs, but still less than the beginning. And here's the oldest one, which was close to a year old, roughly. I think the middle age was like 5 or 6 months, and then this one's about close to a year old. But we found there's almost never a silver bullet where it's a perfect answer, right? Where you're just lucky and you get a solution to all your problems. What we found was brominated DBPs can increase with GAC, and in particular some of these toxic ones we're looking at. This is the halonitromethanes, and if we look at the no GAC scenario where just chlorinating straight up, we don't form any of this tribromonitromethane at all in this situation. Then we start using GAC. Here's our youngest filter. All of a sudden we got 1.9 parts per billion of tribromonitromethane, which is pretty toxic. It's formed when we use GAC. It's not even there if we don't use GAC. And then it's about the same level for the middle age and the oldest. So there's still a few little issues here. And what we think is happening is that when you use GAC to remove your NOM, your natural organic matter, you lower your dissolved organic carbon levels, but your bromide is still the same level. Your carbon's not removing that bromide at all. And so your bromide to DOC ratio has changed and can form some of these brominated DBPs. So what can we do to eliminate these DBPs? I showed the scenario where removing NOM precursors better using GAC. Membranes can also be used to do this, whether they're RO membranes or other kind of microfiltration membranes, nanofiltration. But how to remove the bromide and iodide. If any of y'all can figure this out, you'll probably become a billionaire and you'll save a lot for public health. You'll improve public health a lot. So this is kind of the holy grail to do this cheaply. Of course, you think of anion exchange resins, but it's not cheap. Find a cheap way to do that. This will be great. We got the during scenario, changing the disinfectant. Maybe in a good scenario, I think here is going to UV disinfection within a lower chemical dose for your post-disinfectant for the distribution system. That could be a good way to go. And then another way could be removing the DBPs after they're formed, like your home filters. That's what you're doing. Change your home filters regularly. Though if you use them like I do at home, don't push it past when you're not supposed to be using, because think about the breakthrough you might get. But there's also biological activated carbon at plants. GAC is sometimes used on the indigo plant membranes. Advanced oxidation can be used in different scenarios. I think it's better though to remove things. It's generally better to remove them before they're formed rather than waiting till after. And activated carbon filters can also remove other contaminants like lead, the ones that are certified to remove lead or other things. I can't remember if there's any certified to remove perfluorinated compounds yet, but they can remove a lot of things. And the goal, of course, is clean and safe drinking water. This is what we want. We have. pretty good drinking water, I'd say. I still drink it, right? But I want to make it better. I'd like to improve on it. So we have good water, but let's make it better. And just want to thank you and show a picture of my research group. Here on my postdoc, my new visiting scholar from Campinas, Brazil, and my grad students back here, all these undergrads, enthusiastic undergrads in my group, and my funding sources. Thank you very much for your attention.
Thank you very much, Susan, for this wonderful and very informative talk. We have time for questions. I'm sure there's a lot of you who have some questions for Susan. We have a microphone here.
Thank you. I was wondering, have you guys done any work with like core structure of activated carbon in trying to optimize the different absorption characteristics?
Yeah, we have not, but what we did do, and the royal we, it was Detlef Knabe and Scott Summers, they tested out a lot of different kinds of GAC. What I didn't show is we also had bench skill studies too, a lot of bench skill work. And so they know which ones of these remove, we know now which ones of these remove better. And they do have some idea of the pore characteristics. I don't know the pore characteristics though, but there are all kinds of different kinds of activated carbon, as you know, so, yeah. And by the way, some of them are made from charcoal and some are made from coconut. Have different sources. Those coconut shells, they can burn them and turn them into activated carbon too.
Actually, I was just going to address the question of, this is Judy over here, I'm sorry, it's the other hand. Judy from Wayne State University, Scott Summers, and then also Fred Cannon. Fred Cannon has worked a lot on activated carbon and tailoring it. So those are like the two big names. And then the advisor of Scott Summers, and Lise Weber is the other one.
Hi, I need to introduce myself. So Inge from University of Michigan. Thanks for the inspiring talk. I'm thinking about the area I'm studying, and so I'm thinking the microbial proteins. They're actually nitrogen-containing molecules. I'm wondering, are there any toxic DVPs that form from the reactions with the microbial proteins?
I don't remember if any have been tested for toxicity, and I know that some, like trihalomethanes, have been identified, and bromoforms, is a worthy thing to be concerned about. It's a carcinogen. Bromoform might be a little concerned with the brominated regulated ones, but yeah, different people have looked at microbes and also the intracellular material or something, that looking at microbes as an organic carbon source essentially to form DVPs. And you obviously have them in drinking water, in source waters that you're treating. So yes, they can form DVPs too, but I think there are probably some other things that have not yet been identified. So that's a kind of a new emerging field. I know a couple of people getting in on that. It's right for finding all kinds of new information.
Great talk, Susan. Has anyone looked at, I'm sure they have, but what about alternatives to chlorine as a disinfectant?
Yeah, so that's where I point out the UV. I'm kind of a fan of UV, especially low pressure UV. Medium pressure UV will actually activate organic matter to make it more reactive with post-chlorine and can form higher levels of stuff. So I'm a fan of low pressure UV with a little tiny bit of chlorine at the end. There could be some other things. Some people are using permanganate on the front end as a pre-oxidant sometimes now. I'm also a fan of membranes too, but you can see not a fan of chloramination particularly, just because all the things that have been found, lead being one of them, and iodo-DDPs and nitrosamines and all these other NDPs, and some other issues.
How about peroxide?
Peroxide by itself is not a good disinfectant from what I understand. So I don't know, but UV with hydrogen peroxide can degrade some things. So like if you were the Cincinnati plant, you weren't using GAC, that might be a good way to get rid of some of those industrial contaminants. Some of our emerging contaminants can degrade. It's always important to look at what they're degraded into, right? But a lot of times these things are degraded into things that have less activity or and are more safe. I'd say probably more than 50% of the time they're rendered less toxic, but not always.
So I had a two-part question. Because DBPs, from your talk, wonderful talk by the way, change depending on what's really getting into the wastewater. Are there any policy scenarios wherein water treatment plants in cities are required to work with people like you or people with backgrounds to actually look at high resolution mass spec to see what are the current DBPs that they have and whether their treatment is adequate to support that? And the second, just tag on to that, was point of use filters that people use at home or RO membranes, do they adequately remove all soluble chemicals as well?
Yeah, so let me get the second part of that question, then remind me if I forget the first one. So your home filters, this is a really good question. I almost always get asked this because you want to know. A lot of us have those filters. So if you want to know if they've been tested independently for the things that they're supposed to remove, it'll have an NSF certificational seal on it, and that's not National Science Foundation, but National Sanitation Foundation. It's funny. It'll have the NSF seal on it. That means it's been tested independently by NSF to remove the things that they claim to remove. Doesn't mean they remove everything, just the things they claim to remove. Otherwise, you're taking the company's word for it, basically. But we found that, and I've actually got two undergrads that are almost at the end of a study looking at Brita filters. I wanted to look at other filters too, but too much for undergrads to do. But looking at these other 60 DBPs to see how well they're removed. And overall, it's early, we haven't finished it, but it looks pretty promising. People already know that the regulated haloacidic acids are also very well removed, the trihalomethanes, with activated carbon filters as well as lead, depending which filter, if it's been tested or not. But it's surprising how much activated carbon does remove. So, and if you have RO, well, it's another thing. So if you have RO, if you have a big system that you paid a lot of money for, RO does remove a lot of things. But RO has a general cutoff of about 200 molecular weight, 200 Daltons molecular weight. So NDMA would be small enough to get through that, through RO. That's one of the little issues. That's why NDMA is on that list of 21 priority compounds in the state of California. MTBE gets through. There are a few of these small ones that are less than 200 will get through the RO membranes without completely removed. I think NDMA these days is removed about close to 40-50%. And they're making those membranes better all the time. There's a lot of money going into the RO membrane development, and so they're continuously improving. I think it's only going to get better, but just a caveat, there are a few things that'll get through. And then the other question was, I forgot already.
Just from a policy perspective,
Policy, yeah.
that the water treatment plants in cities, do they have a requirement to have a periodic they do their own analysis, I know, because we work with the local BSA over here, authority, but do they actually have some greater directive to work more closely with, working with high resolution analysis because DBPs keep changing, right?
Right, they do. And so, good question. There is no mandate for them to do that. I'm lucky that a lot of them have agreed to work with me. I keep them all anonymous when I show data, so plant one, whatever, to protect them. But there's no requirement for that, unfortunately. But there are a lot of researchers that are partnering with these plants, and I couldn't do the work I'm doing without them graciously allowing me to get their water, and sometimes at different stages of treatment. Orange County sending me water now, like loads and loads of water in big coolers, and so we're partnering with a lot of places, but they're not mandated. Some of the larger utilities, like one of the ones I think of immediately, one of my collaborators, Stuart Krasner, is at Metropolitan Water District of Southern California, and they wholesale water to Southern California for 20 million people. They're a huge plant, so they do a lot of really nice research. Some of the larger places have their own research people there that do research, but it's only the large utilities in general that do that. So the medium, smaller size ones, unless somebody like me has contacted them, then, there's Toby West, they probably know, they know the regulated DEPs, but they don't know these other ones necessarily. And one other caveat, that the EPA has the UCMR that I mentioned, Unregulated Contaminant Monitoring Rule, and so the nitrosamines were on that, and so all utilities, I think it's, Jody would know, and Angela, over 10,000, I think, people that serve more than 10,000 people have to measure for those contaminants. So we get national data from many, many plants for those, but only for those 25 or so contaminants that are on that list.
Thank you very much. So we actually have a plaque for you from the Renew or to acknowledge you as an innovator.
Yeah, so on behalf of the UB's Renew Institute, it's my great pleasure to present you this plaque. It's called over.